This is week 2 in Genetics 610, and the title is Cytogenetics, Part 2. The learning objectives for this week are to discuss how chromosome analysis is used to detect cancer, to explain applications of fluorescence in situ hybridization, chromosomal microarray analysis, and single gene sequencing, to be able to interpret karyotypes for a range of chromosome anomalies, including sex chromosome abnormalities and other chromosomal variations, and to explain the origin of and give examples of disorders caused by triplet repeats and fragile sites. I want to begin by doing a review on the nomenclature here. I would like you to look at the following: 47XX plus 21, 45XS minus 22, 47XXX, 48XX plus 13 plus 21. I'm going to pause for just a moment, and then I want to move on and to give you the answers of these, because as I said, we do a little bit of review at the beginning of this class. The answers are 47XX plus 21 is a female with trisomy 21. 45XX minus 22 is a female with monosomy 22. 47XXX is a female with 3X chromosomes. 48XX plus 13 plus 21 is a female with trisomy 13 and trisomy 21. I would like you to look at this karyotype and to tell me what it is an example of. I will pause for just a moment, and then I will move on to the answer. This is a karyotype showing a female with triploidy. She has 3 copies of every chromosome, including the X. A technique called FISH, fluorescence in situ hybridization, introduced the ability to highlight individual genes. FISH is more targeted than conventional chromosome staining because it uses DNA probes. The probes are attached to molecules that fluoresce when illuminated, producing a flash of color precisely where the probe binds to a complementary DNA sequence among the chromosomes in a patient sample. Using a FISH probe is a little like a search engine finding the word hippopotamus in a book compared to pulling out all words that have the letters H, P, and O. FISH can paint entire karyotypes by probing each chromosome with several different fluorescent molecules. A computer then integrates the images and creates a unique false color for each chromosome. Many laboratories that perform amniocentesis or CVS use FISH probes for chromosomes 18, 13, 21, and X and Y to quickly identify the most common problems.
This slide is from your core textbook, and it's showing fluorescence in situ hybridization. You can see the metaphase chromosomes are fixed onto slides, and the DNA is separated into single strands. And a single-stranded fluorescent labeled DNA probe binds to the homologous DNA on the slide, and sites of binding are then visualized by using the UV microscope for fluorescence microscopy.
This slide is showing an example of fishing for genes and chromosomes. The fish shows 3 fluorescent dots that correspond to 3 copies of chromosome 21. Each dot represents a specific DNA sequence with which the fluorescently labeled probe forms complementary base pairs.
In looking at the genomic microarray, we see that the resolution of the fish is limited. The smallest visible rearrangements are in the range of about 1 million base pairs. Use of genomic microarrays, which is also called cytogenomics, has vastly increased the resolution analysis. A genomic microarray consists of tens to hundreds of thousands of DNA sequences permanently attached to a solid substrate, each sequence corresponding to a specific region of chromosomal DNA. Hybridization of a test sequence from a patient is used to test for deletion or duplication of corresponding sequences in the test DNA, and it utilizes two methods of detection: comparative genomic hybridization, CGC, or quantitative analysis of hybridization. This next slide shows the process of comparative genomic hybridization. And this process is where the test sample is labeled with red fluorescence and the reference with green. The two are then mixed together. They're hybridized with DNA oligonucleotides, which are bound to a microarray. And equal hybridization produces a yellow signal. Excess test due to duplication results in red, and where you have excess reference due to test DNA deletion, it results in a green signal. This slide is showing in comparative genomic hybridization, it's demonstrating the deletion of a small region on the long arm of chromosome 16, which you can see there with the arrow. Each dot represents the difference in the test versus reference hybridization at a specific oligonucleotide. Most are centered at 0, indicating approximately equal fluorescence. The deletion is indicated by numerous contiguous oligonucleotides, whereas the test hybridization is deficient by one copy compared with the reference. We will be having a guest lecture speak about this technique as well as some of the other cytogenetic techniques in a couple of weeks.
Now let's look at translocations. Translocations are where two non-homologous chromosomes exchange segments. There are two types: Robertsonian translocation and a reciprocal translocation. If you look at the Robertsonian translocation, two non-homologous acrocentric chromosomes break at the centromere and their long arms fuse. The short arms are often lost. This affects about 1 in 1,000 people. The translocation carriers have 45 chromosomes, and they produce unbalanced gametes. If we look at a translocation Down syndrome, we see that 5% of Down syndrome individuals result from a Robertsonian translocation between chromosomes 21 and 14. It tends to recur in families who also have more risk of spontaneous abortions. One of the parents is a translocation carrier, and they may, of course, have no symptoms. Distribution of the unusual chromosome leads to various imbalances. Let's look at an example of a Robertsonian translocation with Mr. P and Mrs. P. Mr. P is a translocation carrier and Mrs. P has normal chromosomes. Mr. P has 45 chromosomes because the long arm of one chromosome 14 has joined the long arm of one chromosome 21. He has no symptoms. Mr. P makes 6 different types of sperm. If we look at A sperm with one normal chromosome 14 and one normal 21 yields a normal child when fertilized. If we look at B, a sperm carrying the translocated chromosome produces a child who is a translocation carrier, like Mr. P. In C, if a sperm contains Mr. P's normal 21 and his translocated chromosome, the child receives too much chromosome 21 material and has Down syndrome. In D, a sperm containing the translocated chromosome and a normal 14 leads to excess chromosome 14 material, which is lethal in the embryo or fetus. If a sperm does not have a chromosome 21, as in E, or does not have a chromosome 14, as in F, a monosomy results, which is lethal prenatally. In reciprocal translocations, two non-homologous chromosomes exchange parts. 1 in 500 people are carriers. usually healthy because they have a normal amount of the genetic material. The translocation breakpoint interrupts a gene, and there may be an associated phenotype. If we look at an example of reciprocal translocation, in such, two non-homologous chromosomes exchange parts. In A, genes C, D, and E on the blue chromosome exchange positions with genes M and N on the red chromosome. In B, a reciprocal translocation has been highlighted using FISH. The pink chromosome with the dab of blue and the blue chromosome with a small section of pink are the translocated chromosomes. Please look at this slide. In detecting chromosomes, laboratory tests to detect or analyze chromosomes can be done on cells sampled at any stage of prenatal development or any age. Many tests are conducted on cells from a fetus or the structures that support it.
I would now like to look at a little bit at cancer. If we look at the levels of cancer, which we see in this slide right here, it takes years for cancer to produce symptoms as more and more cells lose their specializations and divide more frequently than the cells from which they descend. Mutations from single base changes to large-scale chromosomal upheavals drive the disease, accompanied by changes in gene expression, some of which are responses to environmentally induced epigenetic effects. We can see here the diagnosis, the disrupted pathways. We can then look at alterations where we have maybe point mutations, copy number variants, chromosome rearrangements, aneuploidy, or changes in gene expression. An early hint at the genetic nature of cancer was the observation that most substances known to be carcinogens are also mutagens. Therefore, they're damaging the DNA. Researchers first discovered genes that could cause cancer in humans only as far back as 1976. These genes were versions of genes from certain viruses that had been associated with tumors in birds in studies dating from the mid-19th century. In the 1980s and 1990s, research to identify cancer-causing genes began with rare families that had many young members who had the same type of cancer and specific unusual chromosomes. Finding genes in the affected chromosome regions whose protein products could alter cell cycle control led to the discovery of more than 100 oncogenes. An oncogene is a gene that causes cancer when it is expressed, when it wouldn't be in healthy cells, or it is overexpressed. In a sense, oncogenes are inappropriately activated. Family studies also identified more than 30 tumor suppressor genes which cause cancer when they are deleted or inactivated. The normal function of a tumor suppressor gene is to keep the cell cycle running at the appropriate rate for a particular cell type under particular conditions. Most mutations that cause cancer are in oncogenes or tumor suppressor genes. The effects of mutations in oncogenes are typically dominant, and those of tumor suppressor genes are recessive. A third category of cancer genes includes mismatch mutations in DNA repair genes that allow other mutations to persist. When these other mutations activate oncogenes or inactivate tumor suppressor genes, cancer can result. Most DNA repair disorders are inherited in a single gene fashion and are quite rare. They typically cause diverse and widespread tumors, often beginning at a young age. If we look at inherited versus sporadic cancer, we know that cancer is genetic because it is caused by changes in DNA, but it's not usually inherited. Only up to about 10% of cases result from inheriting a cancer susceptibility allele from a parent. The inherited allele is a germline mutation, meaning that it is present in every cell of the individual, including the gametes. Cancer develops when a second mutation occurs in the other allele in a somatic cell in the affected body part. The majority of cancers are sporadic and caused by somatic mutations, which affect only non-sex cells. A sporadic cancer may result from a single dominant mutation or from two recessive mutations in copies of the same gene. The cell loses control of its cell cycle, and accelerated division of its daughter cells forms the tumor. Eventually, tumor cells may contain dozens of mutations that are not in neighboring healthy cells. Germline cancers are rare, but they have a high penetrance and tend to strike earlier in life than do sporadic cancers. Germline mutations may explain why some heavy smokers develop lung cancer, but many do not. The unlucky ones may have inherited a susceptibility allele. Years of exposure to carcinogens and smoke eventually cause a mutation in a tumor suppressor gene or oncogene of the lung cell, giving it a proliferative advantage. Without the susceptibility gene, two recessive somatic mutations are necessary to trigger the cancer. This too can be the result of an environmental insult, but it takes longer for two events to occur than one. Cytogenetic changes in cancer are hallmarks, whether sporadic or familial, particularly in later and more malignant or in invasive stages of tumor development. What this suggests is that the cytogenetic alterations arise because there's defects in genes that are involved in maintaining chromosome stability and integrity, and of keeping the mitotic apparatus functional. Most work was originally done in cytogenetic studies of tumor progression in the leukemias in the beginning. In 1960, the discovery of a tiny abnormal chromosome, the Philadelphia chromosome, by Peter Knoll and David Hungerford was a great breakthrough. It was the first time the cancer was shown to result from a specific genetic abnormality. As cytogenetic techniques improved, by 1973, Raleigh discovered the existence of a translocation between the long arms of chromosomes 9 and 22 in chronic myelogenous leukemia. This translocation resulted in a new fusion protein that was expressed in the cancer cells. This karyotype is showing you the Philadelphia chromosome, which consists of a translocation between chromosomes number 9 and 22, which you can see here. When the CMI progresses from the chronic phase to a severe life-threatening phase, there may be several other cytogenetic abnormalities that might occur, numerical or structural, and one of these is a second copy of the 922 translocation chromosome will occur, or an isochromosome from 17q. In other forms of leukemia, other translocations are common. There are a vast array of chromosomal abnormalities that are seen in most solid tumors. Please watch the video on the Philadelphia chromosome at the following link. Over the past several decades, we have been able to move cancer cytogenetics out of research laboratories and into clinical practice. We're able to recognize today the association between specific cytogenetic abnormalities and certain morphologic and clinical features. And in fact, the World Health Organization has categorized 4 unique AML subtypes according to cytogenetics. Today, conventional cytogenetic analysis is considered mandatory for all newly diagnosed leukemias. FISH is very valuable since it can detect both numerical and structural chromosomal abnormalities. So the cancer chromosomes may be abnormal in number or structure. They may bear translocations, inversions, or have pieces missing or extra. The translocation can increase the expression of a gene, it can turn it into an oncogene, and the duplication increases the number of copies of an oncogene. Deletion may remove a tumor suppressor gene. Retinoblastoma is a rare childhood eye tumor. In 1597, a Dutch anatomist described the eye cancer as a growth the size of 2 fists. In 1889, researchers identified inherited cases of retinoblastoma. The only treatment at this time was the removal of the eye. It's an autosomal dominant disease, therefore there are new mutations. And half of the 1 in 20,000 infants who develop retinitis or retinoblastoma inherit susceptibility to the disorder. After inheriting this susceptibility, they harbor one germline mutant allele for the retinoblastoma gene in each of their eyes. Cancer develops in any somatic cell where the second copy of the RB gene mutates. Inherited retinoblastoma requires 2-point mutations or deletions, one germline and one somatic. In sporadic cases, 2 somatic mutations occur in the RB gene. In fact, the study of RB was the origin of the 2-hit hypothesis of cancer causation, that 2 mutations, germline and somatic or 2 somatic, are required to cause a cancer related to tumor suppressor deletion or malfunction. In this slide, you can see bilateral retinoblastoma. In retinoblastoma, many children have deletions in the same region of the long arm of chromosome 13, 13q14, which led researchers to the cancer-causing gene. In 1987, they found the gene and identified its protein product, which linked the cancer to the control of the cell cycle. Mutations in the RB gene cause other cancers. Children successfully treated have been shown to develop osteosarcoma as teenagers or bladder cancer as adults. Mutant RB genes have been in the cells of breast, lung, prostate, and in acute myeloid leukemia individuals, and in these people, they have never had eye tumors. This may be caused by an expression of the same genetic defect in different tissues. This is a G-banded or GEM610 karyotype from an individual that has retinoblastoma. You can see in this karyotype there are numerous chromosomal abnormalities, including an isochromosome for the short arm of chromosome number 6, of which you can see the arrow. This slide is showing you a photograph of a retina of a child with a retinoblastoma. The white area on the right is the tumor. This slide is of a pedigree showing the dominant transmission of a retinoblastoma. Some individuals have unilateral tumors, whereas others have bilateral retinoblastoma. The father of the youngest affected female is himself unaffected, indicative of non-penetrance. Genes that normally trigger cell division are called proto-oncogenes. They're active where and when high rates of cell division are necessary, such as in a wound or in an embryo. Now, when the proto-oncogenes are transcribed and translated too rapidly, or frequently, or perhaps at the wrong time in development, or at the wrong place in the body, they function as oncogenes. Usually, oncogene activation is associated with a point mutation or a chromosomal translocation or inversion that places the gene next to another that is more highly expressed. Oncogene activation causes a gain of function. In contrast, a tumor suppressor gene mutation is usually a deletion that causes a loss of function. Viruses integrated next to a proto-oncogene can cause rapid transcription when the virus is transcribed. This increases the production of the oncogene-encoded protein, which then switches on genes that promote mitosis, and it triggers various changes. The proto-oncogene may be activated when it is moved next to an actively transcribed gene, and the gland synthesizes the hormone, and the oncogene is expressed. Some examples are in cervical cancer, in anal cancer following an HPV infection, or Burkitt's lymphoma. You can have a translocation or inversion, which places a proto-oncogene next to an antibody gene. So let's look at Burkitt's lymphoma. Here we have a slide that shows that a translocation can cause cancer. In Burkitt's lymphoma, in A, a proto-oncogene on chromosome 8 moves to chromosome 14 next to a highly expressed antibody gene. Overexpression of the translocated proto-oncogene, now an oncogene, triggers the molecular and cellular changes of cancer. In B, you see a child who has Burkitt's lymphoma, which typically affects the jaw. In a type of mutation called an expanding repeat, a gene actually grows as a small part of the DNA sequence is copied and added. One of the first expanding repeat diseases studied was myotonic dystrophy, which begins earlier and causes more severe symptoms from one generation to the next. A grandfather might experience mild weakness in his forearms, but his daughter may have more noticeable arm and leg weakness and a flat facial expression. Her children might have severe muscle weakness. For many years, clinicians thought that the anticipation concept, the worsening of symptoms over generations, was psychological. Then, with the ability to sequence DNA, researchers found that the gene expands. The gene that causes the first recognized type of myotonic dystrophy on chromosome 19 has an area rich in CTG repeats, GAC messenger RNA repeats. The wild type repeat number is 5 to 37. A person with the disease has from 50 to thousands. This is a pedigree of myotonic dystrophy, a triplet repeat disease. In this, we can see how you can explain anticipation. In some diseases, the symptoms that worsen from one generation to the next and have a physical basis. The gene is expanding as the number of repeats increases. So we look here at the first generation where the number of copies was 50 to 80. We go to the second generation, it's 80 to 700. And then when we get into the third generation, it's 700 plus. And you can see at the phenotypes here how they change and how much more severe the disease becomes. Expanding triplets underlie more than 15 disease, and usually a repeat number of fewer than 40 copies is stably transmitted to the next generation, and it doesn't produce symptoms. Let's now look at a triplet repeat disease, which is Fragile X syndrome. It is the most common inherited form of intellectual disability, and it accounts for 3% of autism cases. In the 1940s, geneticists thought that a gene on the X chromosome caused intellectual disability, which was then termed mental retardation, because more affected individuals were male. In 1969, a clue emerged to the genetic basis of X-linked intellectual disability. Two brothers with the condition and their mother had an unusual X chromosome. The tips of one chromosome end dangled like a thin thread. When grown in culture media lacking folic acid, this part of the X chromosome was prone to breaking, hence the name Fragile X syndrome. Worldwide, it affects 1 in 2,000 males, accounting for 4 to 8% of all males with intellectual disability. 1 in 4,000 females is affected. They usually have a milder cases because their cells have abnormal X chromosomes too. This slide is showing Fragile X, where you see the fragile site on the tip of the long arm of the X chromosome, and it's associated, as we've talked about, with an intellectual disability and a characteristic long face that becomes much more pronounced with age. This slide is showing you another child that has Fragile X and some of the characteristics: the prominent long ears, the long face, the delayed speech, large testes, macroorchidism, hyperactivity, tactile defensiveness, gross motor delays, and autistic-like behaviors. This disease should be inherited like any other X-linked disease from carrier mother to affected son, but the penetrance is incomplete. 1 out of 5 males who inherits the chromosomal anomaly have no symptoms, but they transmit the affected chromosome to all their daughters of which half have some mental impairment. And they are called, these males, transmitting males, and their grandchildren may inherit the Fragile X syndrome. The Fragile X area contains 29 or 30 repeats of the sequence CGC in a gene called the Fragile X gene, FMR1. Individuals with the Fragile X chromosome demonstrate the effects of the disorder if this region is expanded to 200 to 2000 CGC repeats. Transmitting males as well as females who have mild symptoms or have affected sons may have a premutation of 55 to 200 repeats. With premutation males, they can have mild neurological problems, and about 1/5 of the women have infertility problems due to ovarian failure. There is a mystery with this disease. A distinct type of disease is seen in the maternal grandfathers of boys who have Fragile X syndrome. The mothers of the boys with Fragile X syndrome often reported the same symptoms in their fathers. Grandfathers were sometimes misdiagnosed with Parkinson's disease due to tremors and their age. Further investigation of the grandfathers has led to the description of Fragile X-associated tremor ataxia syndrome, FXTAS. This has genetic counseling implications since daughters can be counseled that they might pass the condition on to their sons and be offered testing. More is to come on this disorder.